right, let's get started. Should we have a sound check? Can you hear me in the back row? Can you hear me? Yes? Wonderful. I appreciate your being here. I know it's a beautiful day outside, and I know you have alternatives, so thank you for the implicit compliment of being here to learn some more biochemistry or to refresh it. I do not yet have any kind of data about the overall outcome of the exam on Friday. There was a few questions for which I wrote comments that are posted on Icon, and you probably got that email already. But I don't have any data of any kind about the test in general. As soon as I do, I'll share it. Let's go back up. It's time to be aware of things like the mechanics of the final exam. And because we are so numerous, although I know it doesn't look that way today, we have to split between two rooms. And so this room will be one of the two examination rooms. The other examination room is the big one over in the ground floor, the third floor ground floor of Bowen. And it's not labeled as odd one anymore. I'll try to get the actual name off of it. Do you remember what the name is on it now? It's the big one. It has a balcony as well, but I'll ask you to start by filling the lower level first. And I will ask you to try to go to the rooms I've asked you to go to and to bring some kind of name ID. It doesn't have to be the university because we don't have enough room or enough tests to let everybody go where they want. And so please do go to the room that we've asked you to go to. If you have some reason for preferring one room over the other and you notify me of that in advance, I can arrange for, that specifically. But since the two rooms are so close together, I can't imagine it's likely to be necessary for much of anybody. Any other questions about the mechanics of the final exam a week from today? Okay, let's do some reviewing then. So this last week is meant to give you a high altitude overview reminder of a lot of the things we've talked about through the semester. And I hope that a lot of these things will feel familiar to you already. And hopefully what will happen is not only will they be familiar, but the connections amongst them will be that much stronger and more vivid for you. Because in fact, what we've been talking about throughout the semester is connected in many, many ways. And I hope that by having this last week of review, it'll not just refresh the specific facts for you, but it'll help you to see that web of relationships and connections that makes biochemistry such a beautiful subject. And we'll begin with sort of the raw material of life, which is the atoms that are mostly used. There are 6 that are used most abundantly. Carbon, of course. Carbon is the preeminent one. Hydrogen, because there's a lot of hydrogen around. Oxygen, nitrogen, phosphorus, and sulfur. And we'll see when we look at the amino acids how those give their chemistries to molecules of life. These molecules are the key molecules, not the only ones, but the most abundant and key ones for a couple of reasons. One is because of their structure, carbon atoms can do things that nothing else can do. And secondly, because of their abundance. These are among the most abundant elements on the planet. Maybe molybdenum has some wonderful properties, but there isn't enough molybdenum to sneeze at, and so trying to build life around molybdenum wouldn't be very effective. Secondly, they differ in their electronegativity, their tendency to pull electrons in towards the center. And that difference in electronegativity is going to drive the structure of biological molecules, and it's going to drive things like the tendency to reduce or to be oxidized. And so that feeling for the atoms will help you get that sense of it. And so oxygen is very electronegative. It really likes to pull electrons in. And hydrogen is very unelectronegative or electropositive. It tends to lose in the tug of war. And so this fact that they come across a spectrum of tendency to gain or lose electrons is part of how biological molecules get built in useful ways. Not only are there a limited number of atoms that are used, there are also a limited number of functional groups, and we'll see some more of those when we look at some of the other structures. The great majority of the mass of life can be accounted for with just 4 groups of biological molecules. So it's not a huge data set. The simplest group is probably the carbohydrates, which can be thought of as polyalcohols. So they're carbon backbones of anywhere from 3 to 7 carbons with a lot of alcohol groups, OHs, a lot of alcohol groups hanging off of them. They come in a variety of shapes. They either are linear or ring form. Not all sugars can form both, and sometimes it makes a difference which one they tend to prefer, but those are the two principal forms we find them in. Second big class are the amino acids, and the amino acids get their name because they have an amine group, and they have an acid group. So amino acids have a very straightforward name, so they have this split charge or a charged character as an amino acid, and they have the potential for building polymers. Carbohydrates also can build polymers, of course, but we'll see some differences as we go along. The other feature of amino acids is the side chain. So here we have asparagine. This is a side chain off the amino acid. We're going to see the other 19 amino acids also have side chains that make life interesting. Third major group are the nucleotides. In this case, we have a 5-carbon sugar. You can see here it's a sugar because it's a carbon backbone with a lot of alcohols off of it. So there's a 5-carbon that's either going to be ribose or deoxyribose. There's a base of some kind. This is one of the 4 bases, and they're all nitrogen containing. So they're all rings that are heterocycles with some nitrogen in there. So they're going to have a chemistry that's more interesting than just things like benzene or phenol. And finally, the phosphate tails. This is a triphosphate nucleotide. 1, 2, 3 phosphates. These are phosphodiester bonds. So there's on both sides of it, the ester group. And so this is sort of the characteristic overall features you're going to see in any nucleotide. And again, they have the potential to be made into polymers. Fourth group are the lipids. Lipids are not as easily summarized. That is, for the first 3 groups, I can give you kind of a general pattern that you say, okay, that fits, that's an amino acid, that fits, it's a sugar. Lipids have a little more variety in their structures. Just to show you how different some of them can be, here's cholesterol with its rings with a tail, and here's a palmitate up here with its plain old hydrocarbon backbone. I used to say they can't polymerize, but I had a student correct me, so I will defer to that student. They are poorly soluble in water. So if you look at the structure of this, you can see that the carbons and hydrogens are close to each other in electronegativity, so it's not a very polar bond. And we'll be talking about polarity and solubility in just a little bit. And you can think of these really as a lot like hydrocarbons. They have a lot of chemical similarities to the stuff you put into the gas tank. So these 4 groups are enough to give us an amazing variety of things. Now, of those 4 categories of molecules that account for the bulk, not for all, but for the bulk of biological molecules, 3 of them can make polymers. The lipids can get covalently cross-linked to each other, but I'm not going to deal with that in this context. 3 major kinds of polymers, if we want to be more specific. We can talk about proteins, about polysaccharides, and the nucleic acids. They share the features that they are built from subunits. So this idea of building blocks that get made into big things is a theme that connects them. For the most part, they're linear until we talk about the carbohydrates a bit. And because of that, they can have secondary structures, and we'll see what kinds of secondary structures these polymers have. And they can carry information. They can either carry it as a code to be read out in the nucleic acids, or as enzyme specificity in an enzyme, or as the potential for carbohydrate binding in the case of carbohydrates. So this is just your very schematic thing of that. Because when you polymerize monomers, you lose a couple of atoms in the process, it's most appropriate to talk about an amino acid residue or nucleotide residue. It's what's left of the amino acid once it's polymerized. Because we make polypeptides out of 20 amino acids, it really allows for an extraordinary amount of variety of shapes. Those 20 amino acids have side chains that we're going to refresh our memories on that range from very small and hydrophobic to very big and hydrophilic. And so there's a lot of diversity about how you can put together the chemistry of a protein. And that diversity ends up meaning they can do a lot of different things. In life as we know it today, almost all the enzymes are made out of polypeptides. And that's partly because of this huge variety of chemistry available to us through the side chains. In the case of polynucleotides, any given polynucleotide is only going to have 4 bases in it. It'll have A, C, G, and then either T or U, depending on whether it's DNA or RNA. The 2 forms we know of, ribonucleic acid, where the sugar is a ribose, deoxyribonucleic acid, or DNA, because there's an oxygen missing. Very important place, and we'll refresh our memories on that. Polynucleotides or nucleic acids are almost always linear. Very few exceptions to that. And they carry information, especially as a code, but they also carry it to some extent as specificity the way polypeptides can. There's not as much variety of the structural forms in the nucleic acids. Those bases, don't have a lot of differences amongst them. They're either purines or pyrimidines. There's oxygens and carbons and a few, hydrogens and nitrogens. So the variety that's available through nucleic acids is more limited. Polysaccharides, in terms of the bulk of them, that is, if you were to take the mass of polysaccharides in the world, you could say that they're usually homogeneous because the mass of the polysaccharides in the world are cellulose and starch, which are polymers of glucose. We get into things like our own glycoproteins and stuff. They're more complicated, but they're a tiny fraction compared to the cellulose and starch in the world. Polysaccharides make good fuels. They are structural supports for plants, not so much for us. More common amongst the polysaccharides to find branched molecules. They can carry some information in terms of cell-cell contacts, for instance, that the polysaccharides on the surfaces of cells are part of how our immune system knows friend or foe. But it's not its principal function. It's a minority function that's evolved over time. And again, fewer structural forms than the polypeptides because you haven't got the variety of monomers to work from. In fact, you have less variety than you have amongst the nucleic acids. I am not aware of any polysaccharide that has catalytic activity. If someone does, I'd love to know about it. So we have the spectrum from the polypeptides with many enzymes, nucleic acids, a few enzymes, polysaccharides, none at all. Okay. We've also, at various places along the way, talked about the thermodynamics of things a little bit, and the free energy change involved in something. And something that has a reaction such as A goes to B that has a negative free energy associated with it. So this is a minus 20 kilojoules per mole. This is a reaction that will occur spontaneously under the conditions specified. If that were the only thing that happened, life would not be possible because there are a lot of reactions in the body that do not have a negative free energy change. Here's one of them, B to C, has a positive free energy change. But because of the additivity of free energies, if you have linked reactions, so B here is the common intermediate, when we think about A going to C, that reaction overall has a negative free energy change and it can go spontaneously under the conditions that are provided there. And we see this turning up in very broad forms in metabolism where you can really oversimplify what goes on in metabolism in this overall equation, where you start from carbon dioxide, where you have a fully oxidized carbon. And to make things complicated and interesting, you have to reduce it. Reductions are not spontaneous in most cases, so light energy is going to be used to drive that. And here you have a carbon that is partially reduced. This looks like it might be part of a polysaccharide, for instance, or a monosaccharide. going back to where you came from, where you go from this back to a fully oxidized carbon. That's an oxidation reaction. It is favorable. And so the free energy released in that oxidation can be used to do this kind of reaction because of the additivity of free energies. Just a refresher for you on carbon's oxidation states. We saw this near the beginning of the course. We saw it again many times throughout metabolism. The more oxygens that carbon is attached to, the more oxidized it is. The more hydrogens it's attached to, the more reduced it is. So methane is the most reduced form of carbon and has the greatest energy it can be used for. That's the stuff you have in your stovetop, which you can turn on and cook your pasta with it. Carbon dioxide here is the most oxidized form of carbon. That's why you can use carbon dioxide to put out fires. Can't be oxidized anymore, and so it can put out a fire. Most of life occurs in the intermediate range, intermediate oxidation reduction states, and a lot of what we saw in metabolism was taking things either to a more reduced state to do something interesting with it, or oxidizing it to extract energy from it. Any questions? Okay. Feel free to interrupt me. All of these things on our planet occur in the context of living things. And so when we look at cells, when we think about cells, that's where all of the biochemistry is happening. And if we think about the earliest kind of cell that there was, it turns out that these go back a really long time. We can find traces of life 3.5 billion years ago. And these ones that were visible, that have left visible fossils, were the blue-green algae or cyanobacteria. And what was really cool about them then, and what's really cool about them now, is that they can fix carbon, and they can do it through photosynthesis, which is a way of extracting energy from sunlight and fixing carbon. The blue-green algae specifically could do the complicated kind of photosynthesis that releases oxygen. So it is the blue-green algae we have to thank for the oxygen in the atmosphere and the chance to be eukaryotes. I like being a eukaryote much better than I would like being a prokaryote. If any of you have seen this thing about Gliese 351c, or whatever the number is exactly, it's a potentially Earth-like planet around a red dwarf. And if you think about it, as a red dwarf, it's not going to have very high energy photons coming out of it. And so whatever other problems that planet has, it's quite possible that its photosynthesis would not be able to support the kind of photosynthesis that blue-green algae can do to make oxygen. And so it might well be that the life on Gliese, if there is any, has to do it in an anaerobic way. So blue-green algae are among the prokaryotes. They're the eubacteria that have one constellation of biochemistry that goes together, and a related group of prokaryotes, the archaea, which share the fact that they don't have a properly organized nucleus that's completely separating all transcription from all translation. Eukaryotes are more complicated. They probably arose something like 1 billion years ago. The fossils are a little fuzzy. They almost certainly required oxygen to do that. They have a lot more complicated membranes inside than the average prokaryote, although we do know that some prokaryotes can get pretty fancy with their membranes. The double membranes around the nucleus, the mitochondria, and the chloroplast are sort of the hallmark of the eukaryotes. May or may not have a wall. Wide range of shapes, a larger range of shapes, of sizes than the prokaryotes. If you remember, we did that detailed chart showing you that although the prokaryotes can vary over 3 or 4 orders of magnitude, the eukaryotes go all the way up to as long as the neurons in a whale. The complexity of the eukaryotic cell allows complexity of organisms, but that complexity also makes it less robust. And so you do not find eukaryotes in the most extreme environments. If the water's boiling, if it's a sulfuric acid pool, if it's, a mile underground, I can guarantee you that if it's alive, it's a prokaryote, because eukaryotes are just too soft to do that. About the nucleus, which in this case I think, I don't want to swear that I know where the nucleus is in that one. That's a paramecium, but they've stained it in a funny way. But the nucleus is going to be that kind of compact place that provides information for everything else. We talked right at the beginning of this session about the fact that we have life built on the abundant atoms, and water is very abundant, but it's also very unusual. It's a wonderful solvent. And its solvent characteristics are going to be what help drive the structure of proteins and membranes. It's going to be the basis of acid-base reactions. It provides heat capacity. It provides the chemistry that makes biochemistry as we know it happen. And all of that is traceable to the structure of water. And so here you have an oxygen atom in the middle, you have hydrogen atoms to the edges, and these are the unshared electrons sticking out the backside. And so the very big difference in electronegativity between the oxygen and the hydrogen means that these are polar bonds. So you can talk about a partial positive charge, a partial negative charge on these bonds. And if you think about it, the backside is available for bonding too, because it has these unshared electron pairs. And that polarity of the molecule means it can make hydrogen bonds quite easily. The molecule itself is polar, that is, it has a positive end to it and a negative end to it. And so hydrogen bonds look a little bit like this when they're well aligned. Your partial positive of the hydrogen can draw on or be drawn by the partial negative of the oxygen. The degree to which they point straight at each other are going to affect the strength. This gives you a little bit of a sense of the variety of bonds. You've got the hydrogen bonds down here as rather weak and rather long. In biological systems, you see them between hydrogen, oxygen, nitrogen, and sulfur. Ionic interactions are a little stronger, a little shorter length, and of course the strongest ones which hold together the molecules are the covalent ones. These van der Waals interactions are just sort of the surface limits of what makes molecules bump into each other. The hydrogen bonds in water are very abundant. They can make up to 4 bonds per molecule. And because of that extensive network of hydrogen bonds, water has a number of characteristics that it has uniquely compared to other solvents in general, and certainly compared to any abundant other solvents. And perhaps some of the important ones in terms of living systems are things like its heat capacity that acts as a thermal buffer, and it's an extremely good solvent. It can bring in solution a very large range of molecules. And although it's not per se part of biochemistry, the fact that ice floats means life is possible. If ice sank, every time something froze, it'd go to the bottom of the ocean and stay there. So this oddness that the solid form is less dense than the liquid form is an important part of life. And this is just showing you some of the kinds of solvation that go on. Here you have sodium and chloride ions, and the polar character of the water molecules allows it to form shells around each of them. Here's a ball and stick for a glucose. This polyalcohol here obviously has a lot of places it can hydrogen bond to things. And so if a molecule is polar or ionic, water is generally an adequate to good solvent for it. But there are some molecules that are not particularly polar. When we talked about the structure of lipids, that the carbon and hydrogen, close in electronegativity, not a very polar molecule. And as a consequence, it doesn't have any good way to make hydrogen bonds with them. The name we have for these molecules is hydrophobic molecules. They don't really care about water, but they can't interact with it very well. And so if you put one nonpolar molecule into water, there's a thin layer of water around that molecule that is not free to really interact in the same way it would with the rest of the water or with a polar molecule. So it's constrained. Its entropy is reduced because it's not as disordered as it might be. As a consequence, if you put several nonpolar molecules into water at the same time, if they huddle together, there's less water that is constrained, and so this is the most disordered pattern, oddly enough, because the water as a whole is more disordered. This is the basis of the hydrophobic effect. And the hydrophobic effect drives a great deal of structure in biological systems. We're going to see how it contributes to membranes and how it contributes to proteins. To nucleic acids a bit, but we're going to see that nucleic acids depend more on things like stacking interactions. Some molecules like both things. They're amphiphilic. They like water at their polar end, and they don't like water at their non-polar end. They're hydrophobic there. And so the hydrophobic parts will sort out away from water. And they can do that a couple of ways. If you have amphiphilic compounds with one hydrophobic tail, you can make a micelle. If you have two, you can make this very interesting bilayer. It's a bilayer because there's one layer of molecules here and a second layer of molecules there. So the lipid bilayer here is the basis for things like membranes. And it's all based on this sorting away from water in order to get away from that constraint. And because the lipid bilayer has a lipid interior that's hydrophobic and a polar surface, lipid bilayers provide a way to keep things in or out. So here you have a little vesicle, and the stuff that's inside of it is constrained by the fact that they are not soluble in lipids, and so they're going to stay there. And we're going to see ways of getting through that if we have to. Another thing that water can do a little bit, not very much, is dissociate. So water can dissociate into a proton and a hydroxyl ion like this, or H3 plus O. It doesn't do it very much. We're talking about 10 to the minus 7th molar dissociation there. And so at the to try to deal with such small concentrations, the pH scale was cooked up. So the pH of a solution is the negative logarithm of its proton concentration. A pH of 7 means you have an equal number of protons and hydroxyl ions. H plus equals OH minus. Because this is a mass action sort of thing, if you have more protons, you'll have fewer hydroxyl ions. If you have more hydroxyl ions, you'll have fewer protons. And we can reflect that in the pH scale. At 7, you have equal numbers of both. Remember, this is a negative log, so the smaller numbers are giving us more protons. The smaller the pH number, the higher the concentration of protons, the fewer the OHs. Go away from neutrality up to larger pH numbers, negative log, fewer protons, more OHs. So everything above 7 is basic, everything below 7 is acid. Just sort of a reminder of how functional groups react in basic and acid environments, to keep in mind the difference between being protonated and charged. Things like a carboxylic acid, when they're protonated, are uncharged. Things like a primary amine group, when they're protonated, are charged. And you can work through this table on your own just to refresh your memory of that. So let's start thinking about structural biochemistry, structure some of these molecules. And the first major class we talked about were the nucleic acids. We mentioned just now the structure of nucleotides having the ribose or the deoxyribose base. The base carbons are numbered in the plain numbers, the sugars with the primes, and this is the ribose there. The bases are always attached at the 1' of the ribose. And if that's all you have a nucleoside. You have 3 possible pyrimidines to add on. These are the simple single ring with different stuff hanging off the sides. The purines will be a double ring, adenine or guanine. This is just reminding you about nucleotides. Nucleotides can have 1 or 2 or 3 phosphate groups hanging off the side. If they have 3 hanging off, they have 2 high energy bonds like that. If they have 2 hanging off, they have this 1 high energy bond. But a monophosphate has no high energy bonds on it. There's no phosphodiesters in there. And they can be polymerized. And here you have a polymer through the phosphodiester bond. That 5' carbon on the ribose has made a phosphodiester bond to the 3' hydroxyl of its neighbor. And so all nucleic acids have a polarity to them. You can talk about their 5' ends or their 3' ends. They are asymmetric molecules because of the phosphodiester backbone. This particular molecule is a DNA because it has, it's a deoxy backbone. The 2' does not have the oxygen on it. Now, because of that polar character to its backbone, that asymmetric character to its backbone, and because of the bases, there's a possibility to form higher order structures. And certainly the most common one we see is a double helix. And so if you look at this, you can see that we have one of the backbones going 5' down like this to the 3' end, and the other one, the 5' end, goes up like that to its 3' end. So they're anti-parallel. And in the middle of that helix, you have base pairing through hydrogen bonding that holds those bases together. The A to the T, the G to the C. 3 bonds, G to C, 2 to the A and T. Always a purine to a pyrimidine in the standard double helix. There are non-standard base pairings as well. But we also have hydrogen bonds, potential hydrogen bonds, still available. And we're going to remember when we talk about transcription that that plays a role as well. This helix is not a perfectly symmetric helix across it, so you have a wide part to it and a narrow part to it, the major groove and the minor groove. And again, those behave differently in certain contexts. But it means that as you walk along that major groove, for instance, there will be a pattern of potential hydrogen bonds as you walk along that reflects the sequence of that nucleotide, that nucleic acid. The principal form of DNA we see in living cells is going to be the B form. It has the double helix with the phosphodiester backbone like that. An A form that's a little more squished down, it's broader and squatter, is not very common in living things, but is somewhat similar to an RNA-DNA hybrid. A third form, which has a left-handed helix to it, was not thought to be particularly abundant, but it turns out that you find that odd form of DNA in certain segments of DNA that have been methylated as parts of the control regions of things. So it's as if little chunks of this are used to delineate parts of control regions to make it behave differently. RNA molecules can also take on structures. Most generally, RNA is single-stranded, unlike DNA, which is usually double-stranded. But it can fold up. Here's a tRNA that we saw quite recently. And you can see in it regions of base pairing. So quite a lot of this is base paired with some local secondary structure. But overall, it has taken on a complex three-dimensional shape. And so for this tRNA, you can speak about the primary sequence of it. That's the linear sequence of nucleotides. Regions of secondary structure where you have double helix from the base pairing. And overall, a three-dimensional packing that constitutes its tertiary structure unique to that tRNA. The hydrogen bonding provides a way for matching them up. It gives a smooth packing, but it doesn't provide the principal contribution of energy. Some, but not the major part. The principal source of energy for the double helix to form are stacking interactions between the base pairs. That if you think about the footprint of a base pair, it's 3 aromatic rings, and those 3 aromatic rings can sit on each other. If you've ever tried to separate 2 plates of glass that have water between them, that's a physical analog for what stacking interactions are like. Because double-stranded DNA has this complementarity, it makes it a wonderful way to store information. And complementarity lies behind what has been called the central dogma. And it's the fundamental way that most of life works. 99.99 works. The complementarity of DNA, the A to T, the G to C, provides the information for replication. Each new DNA is made from an old DNA. The complementarity of DNA provides for transcription. It templates the RNA molecule. And the complementarity of RNA provides for protein synthesis, but it can't directly because RNA and protein are different things. And so it needs to have adapters. And of course, those adapters are the transfer RNA. In general, information flow is one way. That is, it is from DNA to RNA, from RNA to protein. Bulk of life, that's what's going on. The minor cases are things like RNA viruses, reverse transcriptase, telomerase, but the bulk, the vast majority of information is going to flow DNA to RNA to protein. And this is just a diagram that shows you the complementarity during transcription in a little more detail. And I chose this one because it does have, in the colored version, which you can get offline, of course, separate colors that let you distinguish it. And actually, I think you can see it to some extent in the shades of gray. One strand is going to be a template. It's going to be the thing that is copied. And the RNA polymerase will make a new molecule going from 5' to 3'. So the molecule that's read, the template is read 3' to 5'. It's going to be complementary to the mRNA. The opposite strand that is complementary to the template is sometimes called the coding strand because if you substitute T's for U's, it has the same sequence as the messenger RNA. You make it by copying a template, but it will have the same sequence as the coding strand. Okay. This is just showing you that you have to unwind the DNA for the polymerase to go through and then rewind it afterwards. I keep forgetting that. The code is a triplet code. 3 bases are going to encode the amino acids, 4 possible bases each time, so 64 codons. But those 64 codons are not randomly distributed. It's not just that there's more than one way to say leucine. So here you've got 6 ways to say leucine. It's that everything in this area are all hydrophobic. And so if you make a mistake, it tends to be a conservative mistake. It's redundant. You've got multiple ways to say leucine. It's unambiguous. If I give you a codon, you know what amino acid it codes for. When they did a random draw of this, only one time in a million did they come up with a code that had the beautiful, robust properties that our code has. Okay. I modified this diagram, so now we have dashed lines. I hope it's clearer now in the black and white. All the protein in our bodies had to come from DNA, but not all the DNA in our bodies will make proteins in us. Bacteria, they need to use almost all their DNA. We do a lot of funky things along the way. So if you had 250,000 base pairs transcribed, that initial transcript could be cut and spliced together to make a message that might be only 6,000 bases long. And of that, perhaps only half or less is used to actually make protein. And so for a protein of 1,500 amino acids, those 1,500 amino acids might come from 250,000 bases. And as a consequence, when we look at our genome, only about 1% or so actually makes it into protein. Only maybe a quarter of it is transcribed. And so we don't fully understand what the rest of that DNA is there for yet. But there's a lot of DNA that does not make anything and really shouldn't even be thought of as a gene at all. We have no reason to think of it as a gene. You also have this funny interlocking character of it. I've talked about information going from DNA to make DNA, from DNA to make RNA, RNA to make protein. But it's this interlocking thing. To make DNA, you need both RNA and protein. To make RNA, you need protein and DNA, and so on. And so living systems as we know them now have this kind of complex interlocking character. I think I'm just going to skip on that, but you can refresh yourself on that if you like. Let's just sort of refresh our memories about the amino acids. There are 20 of them, but they fall into a fairly small number of groups. On this page, we have our 8 hydrophobic ones that go anywhere from quite small, like alanine, to quite large, like phenylalanine or tryptophan. And you can see there's almost a sequence of sizes. One rather odd one of proline that gives us kinks and things. Methionine is one of the couple that has a sulfur in it, but it's pretty hydrophobic. Glycine is just sort of small and not particularly polar or nonpolar. Cysteine is not strongly polar or nonpolar either. It's the other sulfur-containing amino acid. And then we have these 6 that are quite polar. You have alcohols. Here's an alcohol and an alcohol. Here's tyrosine, an alcohol smushed onto a phenylalanine. Here we have 2 with amide groups, so you have a carbonyl oxygen here, like that. And you have histidine with its funny ring with its 2 nitrogens in it. So all of these are going to be polar and able to interact with water because they're polar. The histidine, because of its side chain, we'll see can do other things as well. And then there are 4 amino acids that actually have a charged side chain, that are charged under basically any circumstances that we're going to see it in a living thing. 2 that have a negative charge, aspartate and glutamate. 2 that are positively charged, arginine and lysine. Obviously, things that are charged will interact with water very well, or with each other. A couple of these are in between in whether or not they're protonated or not. So both histidine and cysteine can lose or gain their protons in a region close to neutrality. And it's because they can protonate and deprotonate near neutrality that we see them in enzymes over and over again. Cysteine also can do this cute trick here of making a disulfide bond. And so this is a covalent bond that can connect either 2 different peptides or can strengthen an existing peptide by cross-linking within itself. Which brings us to the peptide bond. We talked about amino acids having amine groups and acid groups, your amine group and your acid group. If you take out a water molecule, you can make a peptide bond like this. And because there's an aromatic, some resonance here between these groups, this region here is quasi-planar. It stays pretty much in a flat plane. So this peptide bond, boom, 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 like that, tends to stay flat. The alpha carbon is free to rotate, so these things can rotate around there, but this bond tends to stay flat. And that is part of what contributes to secondary structure when we look at protein structure. I alluded to the idea of orders of structure when we talked about nucleic acids. That idea came from proteins. And I've extended it by analogy to other structures where it's appropriate. But it's first was thought of in proteins. Once you have primary sequence, linear, X, Y, Z, just read it out. There are secondary structures where there's local order that forms, local repeating order. And we'll look at a couple of those, the alpha helix and the beta sheet. You have tertiary structure, where that polypeptide packs in a particular way in three-dimensional space. It might be a mixture of several different kinds of secondary structure, but when it's fully folded, it has one particular packing in three-dimensional space. And then if you have more than one subunit, you have a potential for quaternary structure. So quaternary structure is how the different subunits pack together relative to each other. Both myoglobin and hemoglobin have tertiary structure. Only hemoglobin is going to have quaternary structure. tRNAs have tertiary structure. It's appropriate to talk about the quaternary structure of a ribosome, for instance, where you have both proteins and RNAs in an overall multi-subunit structure. What do some of those secondary structures look like in proteins? Well, proteins have more kinds of secondary structure than some of the other things we've talked about. But there are two that are especially important. There's the alpha helix, and the alpha helix is a helix, that is, it turns as it rises, and it's a right-handed helix, turning as it rises up. And it's held together by hydrogen bonds, as you can see, that go up and down the axis of the helix. So there are hydrogen bonds between the amide nitrogen and the carbonyl oxygen. So there are hydrogen bonds all up and down that alpha helix. The side chains then stick out from this rod-like helix. Obviously, if you had two big side chains next to each other, that might be a problem. Another secondary structure of great importance are the beta sheets. These are, again, held together by hydrogen bonds between the amide nitrogen, carbonyl oxygen, but this time they're side by side. They're either parallel or they're anti-parallel, but the backbone has been pulled out a bit and extended to make a beta-pleated sheet. And here you can see sort of a schematic drawing of an anti-parallel or a parallel pleated sheet. So you have alpha helix, where it turns as it rises. You have beta sheet, where it's laid out flat, side by side. When you look at tertiary structures for proteins, and we'll see implications for other things, certainly in proteins, principally you're going to see the hydrophobic groups on the interior, the hydrophilic groups towards the surface. And in some cases, you're going to see additional things that aren't protein at all, prosthetic groups. So just as a prosthesis, a leg prosthesis is an artificial leg that helps you walk, a prosthetic group helps a protein do things it couldn't do otherwise. And in proteins that have prosthetic groups, they often contribute to the overall packing of the tertiary structure. This is showing you a myoglobin molecule. You've got your heme group here. It's nicely in the interior where it's hydrophobic, and so the water is kept away from the heme iron, and it stays nice and snug. To go from an unfolded chain to a properly folded chain principally is driven by the hydrophobic effect. The hydrogen bonds control the specificity of the packing. They don't control the energy of the packing. The energy that drives it is the hydrophobic effect. The hydrogen bonds produce regions of secondary structure which then can pack together into tertiary structure, but the overall energy of it is driven by the hydrophobic effect. Other features that can appear in protein structures are disulfide ridges or ion pairs. Again, these, oddly enough, don't do as much energy as you might think because the ion pairs were paired with water before they were paired with each other in the protein. But zinc can make a difference. And so here you see a little zinc domain where the zinc binding to those side chains actually does contribute to the overall stability of this protein. We have much reason to think that the sequence of the protein directs the folding of protein, but we don't really know how to predict that yet. We can detect by various physical methods that little regions of secondary structure can form first, and then they can collapse into what sometimes is called a molten globule, where there's a bunch of hydrophobic stuff in the middle, but it isn't really settled down yet. And after it kind of squishes around a little bit, it finds that final packing. into the tertiary structure. In some cases, chaperones actually help that process go forward. In some cases, you actually have to have your prosthetic group or whatever else around to do it, too. Okay. The first protein we really thought about in a serious way was the myoglobin. Myoglobin has a heme in it as its prosthetic group. And interestingly, that prosthetic group can bind oxygen, and that's a key ability for it to do. It can do it in a reversible way, and this curve we're seeing here, we'll see in a number of settings, is a hyperbolic curve. It's a sign of a simple binding relationship that doesn't have anything very fancy about it. It's just, it binds or it's not. And we can talk about its affinity for things. And here we're going to get into something we see many times, which is it's hard to talk about the absolutely full thing, so we often talk about the half-full thing. And we can talk about fractional saturation. It's P50. It's half saturated at 2.8 torrs. So it has a pretty high affinity, a very small P50, and it's time to say goodbye. You're welcome to look at the rest of these slides on your own. I think we're pretty close to the end of those.